95% of drug development in Alzheimer's disease is really focused on amyloid and trying to target that amyloid pathway in some way or another, either altering the way that amyloid is formed from the precursor protein, amyloid precursor protein, or trying to get rid of the amyloid from the brain in some way or another. And in fact, the initial studies in mice were conducted in the mid-1990s with some of these approaches. These are transgenic mice. These are mice that carry familial genes for Alzheimer's disease, so the genes that cause Alzheimer's disease in people where it's inherited in families and people develop it in their 30s and 40s. And those genes lead to the overproduction of amyloid. And you can see in these mice, at 18 months of age, without the treatment, you can see in the middle there all the deposits of amyloid in the brain of these mice. The treatment that was brought forward at this point was a vaccine treatment. So this was injection of a fragment of the amyloid protein. The idea was that people would develop antibodies to that protein, and that would then start to clear it from the brain. And in the mice, that worked fantastically. You can see there at 18 months of age in panel C, the mice that had had the vaccine almost completely clears the amyloid protein from the brain. And I think those of us who were researchers in the field thought, well, you know, we better find something else to be interested in. This is the end of Alzheimer's disease. It's all cured. Unfortunately, translating things into humans is kind of not quite as straightforward as that. Because of all the excitement around the animal studies, this moved very quickly forward into human phase 1 and phase 2 trials. But these trials were stopped because of aseptic meningitis in about 6% of participants, some of which led to deaths, and the trial was halted. But because this was obviously an active vaccine study, once the treatment's been given, you can't ungive it. So a number of these people were then followed up until death. And the brains were examined. And in a very, very important study, Clive Holmes and colleagues from Southampton reported in The Lancet back in 2008 the outcomes for some of these individuals that had come to post-mortem. And the interesting thing is that exactly as happens in the animals, actually the vaccine did clear the amyloid. 80% of the amyloid disappeared from the brains of people who'd had the vaccine treatment. Unfortunately, they still continued to progress at exactly the same rate as they had, as if they had still got amyloid in their brains, and they died with end-stage Alzheimer's disease. So the treatment actually got rid of the amyloid, but it didn't improve the clinical outcome. And I think this was a really important, but actually largely ignored finding that probably suggests that, I'm not saying that amyloid isn't important, because I think there's lots of evidence that suggests that it is, but probably it's not sufficient just targeting amyloid, or, as has also been put forward as a hypothesis, that by the time we try and treat it, there's already so much amyloid in the brain that it's actually closing the stable door after the horse has bolted, and that it needs to happen at a much earlier stage. So largely because of the adverse events of the active vaccines, the next sort of stage in the development of this kind of line of treatment was to try and develop passive immunotherapy, so to actually give the antibodies directly rather than to give an active vaccine. Obviously, this has kind of potential safety advantages because you can stop the therapy if there are adverse events. And the tolerability is generally a lot better. There were still some adverse events. It didn't cause a lot of mortality. It did cause some brain inflammation, which is reversible when the treatments were stopped. So from a safety point of view, this was a lot better than the active vaccine. But unfortunately, despite an awful lot of investment in this area, a lot of trials, a lot of very large, expensive trials, so far they've all failed. But I kind of think that if you go back to the Clive Holmes study, it's perhaps not that surprising because the Holmes study was suggesting that actually the treatment was doing exactly what it was intended to do. That just wasn't enough to improve the outcome of the disease. So it kind of feels not at all surprising that if that's what the science is telling us, that it's not sufficient, that actually developing more therapies targeting the same thing in a direct way probably isn't going to be the answer. So what else do we do? I mean, I showed you on the slide the tau pathology in the brain, Tau is probably a good target. It develops a little bit later than amyloid. If you look at correlation studies from people who've donated their brains at post-mortem and where there's been correlations between the severity of impairment and the pathology, Tau correlates much better with the severity of pathology, with the severity of clinical impairment than amyloid does. Because it develops a little bit later, it might be a better thing to target during the actual disease process. We are now beginning to have some trials looking at Tau. Unfortunately, the Tau therapeutics product has recently failed in clinical trials. There are a number of studies looking at Tau antibodies that are beginning to come into clinical trials and are looking promising in early phase development. So that might be a more promising avenue. I think what's beginning to be encouraging is that we are also beginning to see a number of other things. We're beginning to see symptomatic therapies being looked at again. I think for a long time the thought was, well, you know, we've got cholinesterase inhibitors, we've got memantine, what we now need is a disease-modifying therapy. And whilst I think it's totally true that a disease-modifying therapy would be amazing and would be a massive step forward, actually, if we could double the impact of a symptomatic treatment by combining therapies, that would also be a very, very substantial gain, especially for people who are already in the moderate stages of the disease. And actually, an awful lot of people aren't currently diagnosed until they get to the moderate stages of the disease. So for those individuals, symptomatic therapies such as 5-HT6 agents would also be a major advantage. We're also beginning to see some trials looking at people in the very, very early stages or even pre-disease. And an example of this is the Takeda study mentioned on the slide there, where that study is actually looking at people who are healthy older adults but have genetic risk factors for Alzheimer's disease, where they carry the ApoE4 gene, which isn't a direct cause of Alzheimer's disease, but if you have one of those genes, it increases the risk about 4-fold. So targeting those individuals and trying to prevent the early stages of cognitive decline. So I think that's a very brave study, but I think it's probably the right place for studies to be if we're looking at some of the early pathological changes and targeting them. And I suspect that will be a trial design we see a lot more of in the future. So I think that, why I said the jury's out, well, we've already got cholinesterase inhibitors. That's thanks to neuroscience. We've invested a lot of effort in amyloid therapies, so far unsuccessfully, and I actually believe the science is telling us that it's very unlikely that those approaches are going to be effective as single therapies. We're beginning now to branch out and follow up on our understanding of things like tau pathology, but the jury is still out on whether those are going to develop effective therapies and whether they're going to deliver benefits for people within the next 10 to 15 years. So I think it's an exciting time. There's a lot more work, there's a lot more diversity, but it's still not proven that neuroscience will deliver clinical therapies for us that are effective.